Hello everyone, welcome to Practically's NEET Bio class, and today we're going to learn about the cell. Okay, so today we're going to learn about cell, what cell is, and what is basically cell theory, where does the cell come from, okay, and who discovered it, where does the history, and how it works, how the cell works, what are the different kinds of cell. Okay, and apart from that, we're going to learn a little bit about prokaryotic cell. Okay, that is the primitive type of cell. And with that, we'll close today. In the next classes, we'll be learning about the developed cell, that is the eukaryotic cell. Okay, so let's get started. Before that, I'd like to talk about us. Okay, we are from Practically, and it is India's first experiential app. You get lots of simulations, videos, which runs for 6th grade to 12th grade, and we have regular courses running on the app as well as on YouTube also. And you can follow us on social media from Instagram and on Facebook. And do not forget to like, share, and subscribe to our videos on YouTube. So now, if you have any doubts or anything, okay, so you can put it on the chat box and I'll be sure to look at it and I'll try to solve your doubts as well. Okay, hi Neha, good evening. Welcome to the class. Okay, so let's get started. Now, before I start talking about what cell is and what is the definition of cell, how many types of cells there are, etc. Before that, we'll talk a little bit about the history of cell. Okay, so basically, there was a person called Roger Bacon. That is from where it started. So that is from where people started to know about cell. So what Roger Bacon did, he was a British philosopher, and he had a lot of ideas on optics, on different kind alchemy and all. He was basically a very brilliant person. Okay, and he was the one who actually invented the magnifying glass. Okay, for scientific purposes. He also had a lot of views on how the world is and how it is around a circle shaped area and how we can go around it and everything. He was basically a brilliant person, and he was the one who identified a magnifying glass. So why magnifying glass? Because only if there is a magnifying glass, then only we can see the cells, right? Cells cannot be seen with the help of naked eyes. We need something to see it. That is where it started. He discovered the magnifying glass. Done. Now then, in another some 200-300 years after that, a father and son duo called Hans and Zacharias Janssen, they invented microscope. Okay, magnifying glass was done by Roger Bacon. These people, they were working in optics, okay, in the field of optics, and they discovered microscope, with the help of which you can actually see small things, worms, insects, etc. They did not feel like, they did not think that it will be a very marvelous invention or anything, but it actually worked very nicely. Why? Because it is the help of that only that Robert Hooke finally he discovered cell. Okay, he was the one actually who discovered cell. So he actually saw some dead cork, cells cork, as in the bark of a tree. That cell he took. Okay, that is actually a dead cell. Okay, and he actually placed it under, cut it in a thin slice, and he placed it under his microscope. Okay, and what he saw was this diagram. Can you see? This diagram is, this is what he saw. Some small rooms or boxes he saw. Okay, so it was Robert Hooke who actually invented cells. Okay. Now, after that, many other people came. One of them was Anton van Leeuwenhoek. What he did was, he observed a living cell. Because Robert Hooke only saw a dead cell. It was Anton van Leeuwenhoek who actually observed a living cell. Okay, now done with the discoveries. Now, a little bit I'll talk about the different kind of microscopes, because till the time when actually a magnifying glass was discovered by Roger Bacon, that was in the 1200s, 1300s, till now we are in the year 2021. The field of microscopy has jumped a lot. Okay, because previously people used to use light microscopes and all. Nowadays we have such immensely powerful microscopes such as electron microscopes, that not only can we see the structure of a cell, we can actually see what is inside the cell. We can actually see how the reactions go on. We can see smaller chromosomes and everything, DNA and everything. We can actually see that very nicely. Okay, and the resolution will be so great because we have something called the electron microscope. Okay, and electron microscope is of two types. There is transmission electron microscope, that is STEM, and there is scanning electron microscope, that is SEM. Okay, so even in the field of microscopy also, for the past, like in the so many decades we have jumped, and still we are still working on even better microscopes than the electron microscope. Okay, so now we'll talk a little bit about the cell theory. So basically, cell is what? Cell is the structural and functional unit of life. Okay, so it gives us structure also. It works on a function basis also. So cell is basically the structural and functional unit of life. So now a lot of people had a lot of opinions about cell. Okay, so in those opinions, there are three opinions which actually were conjoined together, and a version of cell theory was given. Okay, so it started with a person called Matthias Schleiden. Okay, so he was basically a botanist. Okay, in the year 1838, he actually concluded that all plants are composed of cells. Okay, simultaneously, a zoologist called Theodor Schwann, okay, he concluded that all the animals are also composed of cells. Okay, now there was another person called Rudolf Virchow. He said that cells come only from another cells. Okay, so together basically the cell theory was given basically by Schleiden and Schwann, that is Matthias Schleiden and Theodor Schwann. Okay, and then Rudolf Virchow, he gave a little bit of his own like ideas. But the first of all the cell theories, there is also another modern cell theory also, but the first version of cell theory was summed up in these 3 points. The first thing is all living things are composed of one or more cells. Okay, cells are the organism's basic unit of structure and function, and cells only come from existing cells. Okay, so these are the 3 points when it comes to cell theory. Okay, now this was the first draft. Consider this as the first draft. Okay, after this, a lot of, since this was only done in the 1800s, after that so many years have actually passed, and scientists had a lot of their own opinions as well. As I said, when the cell was actually it was invented, after that a number of people had a number of opinions. Okay, so as time passed, people had started putting their own opinions one after the other. Now we actually have a modern version also. Okay, so what does the modern version of cell theory say? That energy flow occurs within cells. There are those 3 points also. That is the original cell theory. People have added their own things. Okay, so the first thing is energy flow occurs within cells. Okay, hereditary information, that is DNA, is passed from cell to cell, and all cells have the basic chemical composition. Okay, carbohydrates, proteins, lipids, etc. Those are the biomolecules. Okay, so these are the 3 points which were added later. Okay, apart from those 3, these were the 3 points which were added later. Okay, let's see who and all has joined. Okay, hi Nanda, hi Manoj, hi Sagar. Okay. Yes, Sagar, that is what Rudolf Virchow he said. He says that cells can only come from a mother cell. One cell can give birth to another cell. Cell just like that, it does not come out. It is not magic or anything. So cells, one cell can come from another cell. Okay. So, second. Okay. Now what? Now I'll tell a little bit, we'll discuss about what are the different kinds of cell we see. Cells can actually be divided in n number of types. Okay, there is a unicellular and multicellular, there is prokaryotes, eukaryotes. Okay, then different shapes of cells and everything. There are a lot of cells. Okay, mostly if we consider, even if we don't consider the dead parts, okay, the dead cells, if only we consider the living cells as well, there are n number of shapes, n number of types and everything. So organisms show variety in cell number, shape, size, etc. Okay, so when it comes to the number of cells, there are actually 2 kinds. The first is unicellular cells. So uni means 1. Whenever you hear the word uni, that means 1. So one celled. Okay, so one celled organisms are basically microorganisms. Okay, it can be bacteria cells, primitive cells such as bacteria. It can be a bit developed ones such as yeast, amoeba, paramecium and all. So all of these are organisms, be it paramecium, be it amoeba, be it bacteria, yeast, okay, all of these are unicellular. They are one-celled organism. Okay, they do not have hands, legs, other parts and all. They don't have. They are just one cell. All of their metabolic activities, all of their life, everything depends. Everything is in that one cell only. They live. Okay. Now, when it comes to multicellular, that is what we are, multicellular organisms. We are higher organisms. No, we call ourselves higher organisms. Why? Because we are multicellular. Okay, so more than one cells. We have different kind of, if we consider humans, we are multicellular. We have different kinds of cells. It's not even the number of cells. We have different kinds of cells as well. So when you talk about, we have a stem cell that is an origin. From there we have muscle cell, fat cells, bone cells, blood cells, nervous cells, epithelial cells, immune cells, sex cells, so many different kinds of cell in one body we have. Okay, so different kinds of cell in one body. It can be similar kind of cells in a body also. It can be different kinds of cells in one body also. So any organism which has more than one cell is known as a multicellular organism. Okay. Now, shape of cells. Shape of cells are actually, the thing is, there are, it depends. Okay, a shape of cell, you cannot just say that plant cell is rectangular in nature, or what to say, the animal cell is oval in nature. No, shape of cell actually varies. It varies from in different organisms, it is different. Okay, when I showed you this picture, this is the number of cells we have in our body. See, you can actually see that how different each and every shape are. Can you see, nervous cell is like a string, a long string. Red blood cells are a bit oval in nature. Muscle cells are straight line like. Okay, so each and every shape is different. So a shape, there are shapes such as, there are organisms such as amoeba and all, okay, who have very irregular shape. The thing is, they do not have a proper shape, okay, they keep changing their shape. I have, okay, so they keep changing their shape. That's why they are called pseudopods. So this pseudopod thing actually keeps changing. When they move, the shape of the amoeba keeps changing. Okay, so these are like irregularly shaped cells. Okay, then we have white blood cells or something like that. Okay, they can also change their shape. That was microorganisms, a lower one. Okay, we as human beings also, we have WBCs which can actually change its shape. Okay, so when that amoeba is a full-fledged organism, but for us WBCs is just a part of us, but they can also keep changing their shape. Okay, and then there are other shape cells, such as this is a neuron, okay, or a nervous cell. It is so long, it is one of the largest cells, okay, in terms of the size, it is one of the largest cells, and it in itself only it has different shape. See, here it is irregular, here it is straight, again then irregular. So they are actually a very unique kind of cells. Okay, now again, when we talk about the size of the cells, when it comes to size of the cells, cells can be very little, very minute. Okay, micrometer or micron we can measure it, such as bacterial cells. Okay, these cannot be visible with an eye. Okay, you need a proper highly resolution, a kind of microscope you need. Okay, other than that, this is one of the most smallest cells you can actually find. But there are also cells such as an ostrich's egg. This is an ostrich's egg. Okay, which is actually one of the largest cells in the world. Okay, and it can actually, it is one cell only. It is not that it is a number of cells inside. It is just one cell, and it can actually measure 170 millimeter into 130 millimeter. Such large it is. So size of the cell can range from 0.1 to 0.5 micrometer or microns, okay, to such big shapes. Okay, that is 170 millimeter into 130 millimeter. This is such big shape. So a cell can be small also, a cell can be large also. Now in our body, the longest cell in our body is the nerve cell. Just now we saw this is the largest cell in our body. Okay, again, if you have any doubts or anything, okay, please let me know so that I can help you. Okay, next, the types of cell. Okay, so cells can actually be divided into two types based on the development and everything. Okay, so there is prokaryotic cell. These are primitive cells, and there is eukaryotic cell, that is developed cells. Okay, now prokaryotic cells, as in they have they are actually, they do not have a true nucleus, they do not have a lot of organelles. Okay, they have very few organelles, and they are covered by a capsule and everything. So these are primitive cells. Basically, bacteria and blue-green algae and all are prokaryotic cells. Now, eukaryotic cells are the more developed ones. They have proper shape. They have proper organelles. They have a true nucleus. Whenever you say eukaryotic cell, means they have a true nucleus. Prokaryotic cells, they do not have nucleus. They have a nucleoid, whereas eukaryotic cells, they have a true nucleus. Okay, that is why they are called eukaryotic cells. Okay, now we'll be discussing about the prokaryotic cell. Today we'll only be considering the prokaryotic cell, whatever is there in a prokaryotic cell, although not a lot of things are actually there in a prokaryotic cell, not a lot of organelles or anything. We'll just discuss, and in the next classes we'll put more effort into the eukaryotic cell. Okay, so the first is the prokaryotic cell. They were first form of life on this planet. That is why right? Prokaryote. Why primitive kind? Why do we call that? Because it is the first form of life on the planet. They are smaller and simpler than eukaryotes. Because why? They do not have a lot of organelles. They do not have a proper nucleus or anything. That is why they are smaller and simpler. Okay. Prokaryotic cells, they lack well-defined nucleus, and they do not have any membrane-bound organelles such as mitochondria and all are not present in the prokaryotic cell. They do not have a well-defined nucleus. Why? They have something called a nucleoid. Okay, that is not a proper nucleus. There is a genetic material, okay, and it's just floating in the cytoplasm. It's not covered. Okay, there is no nuclear envelope, or there is no nucleolome and everything. That and all is not there. So it is just a DNA of prokaryotic cell consisting of single chromosome. Okay, and it just floats in the cytoplasm. Okay, and for example, bacteria, archaea, blue-green algae, these are the organisms which are kind of prokaryotic cells. Okay, let me see. Hi Taresh, welcome to the class. Okay, so now what are the things I've been saying? Okay, that prokaryotic cells do not have anything. What are the things which are actually there in prokaryotic cells? Firstly, they have cell membrane, they have cell wall, and they have cell capsule. Okay, so the first thing they have is cell capsule or an envelope. Okay, then they have cell wall, they have cell membrane, then there is the cytoplasm inside. These are all the coverings. Okay, these are all the covering of the cell. Okay, then there is cytoplasm, that is the jelly-like structure in which the organelles are. Okay, ribosomes, this is there because they make protein and everything. This is for protein synthesis. Genetic material, that is DNA, they have. Okay, that is just a free living DNA in the cytoplasm they have. Plasmids they have. Okay, and flagella, pili or fimbri. Flagella is basically for movement. Pili, fimbri can be for movement, can be for contact between two bacterial cells and all. Okay, that can be there for pili and fimbri. Okay, so this is how the cell looks. Okay, these are the components of the prokaryotic cell. Components of prokaryotic cell. So we'll start with the outer covering, that is the cell capsule. Now please understand that this thing, cell capsule, is only present in prokaryotes and not in eukaryotes. Okay, so what is this? This is basically a sticky outermost layer of the capsule made up of basically polysaccharides, sugars. So the outermost, sorry, 1 second. So the outermost covering of the cell is known as the cell capsule. Okay. Now the capsule helps what? The prokaryotes to cling to each other and to various surfaces because it is sticky, right? It is sticky in nature and it clings to the surface or holds on to different different cells or different bacteria or different surfaces. Because of the sticky nature, this is how it works. Okay. And it also helps preventing the cell from drying out because it is sticky in nature, it is mucousy in nature. It doesn't let the cell to dry out. In the case of disease-causing prokaryotes that have colonized the body of a host organism, a capsule or slime layer may protect against the host immune system. Now, think about this. There are lots of bacteria which actually harm us, such as Staphylococcus aureus, Staphylococcus hemolyticus, and all that. Okay, these are all bacterial cells. Now, what they do, they harm us. Maybe some living organisms, such as humans and all, what they do, they somehow get into our body. Okay, but our body is made up of a lot of things, right? Our body is made up of a number of enzymes and hormones and other cells, T cells, B cells, and everything, immune system and all that we have. So how do these cells, these small bacterial cells, how do they save themselves? They save themselves because they have this cell capsule. Okay, this is quite protective in nature, and they basically save the body, save the bacterial body from harm. Okay, so it is a protective function. Okay, so that is the cell capsule. Now comes the cell wall. What we are doing is we are going inside from the outer part, we are going inside. So the second barrier which you see in a cell, prokaryotic cell is the cell wall. Okay, so this is quite stiff in nature and it is located just underneath the capsule. Okay, and what is it made up of? It is basically made up of peptidoglycan. Okay, something called peptidoglycan. Okay, with what is that? It is a mixture of carbohydrates and proteins. So it is a layer, a carbohydrate and protein based layer. So what happens? This peptidoglycan, there are basically two kinds of bacteria. Okay, there is a gram-positive bacteria and there is a gram-negative bacteria. How do we find out what is a gram-positive and a gram-negative bacteria? That is done by something called gram staining. Okay, so how do we find out when it is a gram-positive bacteria, they have only this peptidoglycan layer. Okay, so a gram-positive bacteria will only have the peptidoglycan layer. When it comes to the gram-negative bacteria, they have a smaller peptidoglycan layer and on top of that, they have an outer membrane also. Okay, so when it is gram-positive, it is only the peptidoglycan layer. When it is gram-negative, it is peptidoglycan plus outer membrane. Okay, so when you stain a cell, when you go for gram staining, you stain it, as in you try to color it with two kinds of colors. Okay, there is a violet color and there is a pink color. So what happens in gram-positive bacteria, due to the presence of this thick, gram-positive has a thick peptidoglycan layer, no? So they retain the crystal violet color. Okay, because they have taken all the colors. Okay, peptidoglycan has accepted all the colors. So gram-positive bacteria is violet. When it comes to gram-negative, okay, they do not retain the crystal violet color. They do not retain it because they have an extra layer, right? You can see here, they have an extra layer here. Okay, here, they have an extra layer here. So what they do, they do not take that violet color, they do not take it. They can, if they get washed, they throw it away. So because of that, they do not obtain that violet color. Instead, they just retain a light pink color. Okay, so that is why when you gram stain it, when you stain a cell, if it's a gram positive cell, it will be violet in color, somewhat like this. Okay, and if it's gram negative, it will be pink color, like this. And all of this because gram positive has a proper peptidoglycan layer and gram negative has a peptidoglycan, a smaller peptidoglycan and an extra membrane. Okay, I hope you understood it. If you did not understand it, please just let me know and I can very well go back again and make you understand. Okay, now the next membrane, okay, till now we have discussed the capsule and the cell wall. The next thing is the plasma membrane. Now remember, plasma membrane is also present in a eukaryotic cell as well. Okay, we'll discuss more about the plasma membrane, more in details about the plasma membrane structure and all. We'll discuss it in the next class. Okay, it will be way more interesting then. Okay, so what happens? This is the plasma membrane. Now, what is plasma membrane? It is made up of a phospholipid, a lipid composed of glycerol molecule attached to a hydrophilic phosphate and two hydrophobic fatty acid tails. So basically, it looks like this. Okay, so what is it? It is a lipid composed of glycerol molecule attached to a head. So this is the head. This is hydrophilic. It attracts water. And there is the hydrophobic tail. These are fatty acids. So they kind of repel water. So they are hydrophobic. Okay. Now the phospholipids of eukaryotes or other bacterial membrane are two layers. Okay, they form a phospholipid bilayer. So how it looks is, it looks somewhat like this. Okay, so the hydrophilic layer will be up and down in the ends, and the hydrophobic layer will be in the middle. Okay, now there are plasma membranes of archaebacteria. They have a bit different. Okay, they have phospholipids are joined into single tail, and they have a monolayer. This is lipid bilayer, right? This is bilayer, as I said, upper and niche. This is a monolayer. Okay, it just is like this. This is in some archaebacteria. Okay, even in bacteria and eukaryotes, it is bilayer. Looks, I'll just draw it once more with a clear picture. It is somewhat like this. Okay, so we'll discuss more. This is just the lipid bilayer. We'll discuss more. There are some proteins, there are other things also, but we'll be discussing plasma membrane in proper details in the next class for the eukaryotic cell. Okay, now comes the cytoplasm. Cytoplasm is the jelly-like structure. Okay, it is where the genetic material floats. It is basically a jelly-like structure and it is there inside the cell. And on it, the genetic material, the ribosomes and all, everything floats. That is the cytoplasm. Okay. Now, what organelles are there? There are not a lot of organelles, but one of the most important organelle there is the ribosome. Okay. And ribosome is required for protein synthesis. They have to synthesize protein, and the metabolic activity and everything is controlled by the protein synthesis and all. So they have to, like, for the survival of the organism, the functioning, structuring and everything, you need a ribosome, you need protein synthesis, and that is controlled by the ribosome. And now ribosome actually has, even in eukaryotes, ribosome actually has two parts. Can break up the ribosome into a smaller subunit. This is a smaller subunit, and there is a larger subunit. Okay. And you can calculate it with unit S, that is Wedberg units. Okay. So prokaryotic ribosome is 70S, that is 70 Wedberg units. Now the smaller section is 30S, made up of 30 Wedberg units, and the larger portion is 50S, that is 50 Wedberg units. And together they make 70. It does not follow proper rules of mathematics, because 50 plus 30 is actually 80, but it comes up to 70 Wedberg units. That is the prokaryotic ribosome. Okay. Now the next thing is the genetic material. Just before beginning only, I had actually discussed that the genetic material of the genetic material of prokaryotes are actually, they are not developed. It's not nucleus. It's not true nucleus. Okay. So basically they have something, see this violet zigzag thing you can see, right? That is the nucleoid. It's kind of double-stranded DNA. Okay. Circular double-stranded DNA. And it has all the genetic information, the DNA, it has all the genetic chromosomes and all they have. And these genes are required for growth, survival, reproduction and all. Because they have to, even if they are like unicellular, they have to grow, right? They have to divide, they have to reproduce. So that kind of function is helped by the nucleus. Okay. Now it looks like a mesh of string in the middle of the cell. See how it looks. Looks like a mesh of string like this. Okay, it just floats in the cytoplasm and it looks like a mesh of string. Now, generally, DNA is spread throughout the entire cell, okay, and is readily accessible for the transcription. Okay, so there are no envelopes and there are no coverings on everything. Okay, so they are just there floating inside the cell, total cell they have, and they are floating inside that. And they are readily available for transcription, as in what happens? Transcription is what? DNA gets converted into RNA. Okay. And then ribosome works. This RNA gets converted into protein. Okay. And sometimes when you want to actually see the DNA, okay, you can actually condense it in a part of the cell, and this darkened area will be, when you see it in the microscope, you can see it in the darkened area, you can see the DNA, that is the nucleoid. Not the nucleus, it is not a true nucleus, it is known as the nucleoid. Okay, Siddharth, okay, is asking what is the role of RNA. RNA is there for, it is the base for protein synthesis. It is because the You need the protein, right? And the ribosomes can have everything. They'll work only. Ribosomes, what they do, they take mRNA, okay, and they convert it into a protein. That is the base. The template is RNA. And RNA also is a kind of genetic material. Like DNA, RNA also is a kind of genetic material. Okay. Now they have another structure. Not everyone, okay, they have, some of the bacteria actually has something called plasmids. Okay. So what is it? It is the small DNA, the small pieces of DNA, because the whole structure of bacteria is covered with this thing, no, genetic material. It's just floating there. So there are also small DNA particles, and these smaller DNA particles are known as plasmids. Okay, so they are mostly circular in nature. They look somewhat like this. Okay, and can be transferred from one prokaryotic cell to another through the pili. Okay, so they can be transferred, okay, during reproduction and all that. Okay, that they can actually be transferred from one bacterial cell to another. Okay, and these are also very good. They work very good as vectors. Okay, in genetic engineering, okay, you can use these plasmids as vectors to carry some genes, okay, such as antibiotic resistant genes and all that. They can actually carry these plasmids, actually carry these genes, specialized genes, from one cell to another. That is why they can be very good vectors in the field of genetic engineering. Okay, what else is there? There are the appendages. Appendages as in how they move, okay, how they contact with each other. Okay, so those two of the three appendages they have, that is the flagella is basically used for movement. Appendages here can be pili and fimbriae. Okay, so prokaryotic cells often have appendages or protrusions from the cell surface, that extra portions from the cell surface that allow the cell to stick to the surfaces and move around. Okay, or transfer DNA to the other surfaces for moving or for sticking or for transferring DNA, plasmids and all that. They have these appendages or the protrusions. So out of them, there is the thin filaments called fimbriae, okay, or fimbria. They are used for adhesion. Adhesion basically means for sticking. Okay, just before I started the prokaryotic cells, I told you they have a sticky nature. They have to stick somewhere. They have to, if they want to depend on someone, they have to stick somewhere. That kind of work is done by the fimbriae or fimbriae. Okay. Now these are smaller appendages. There are larger here, you can see. These are the fimbriae. Now there are larger appendages such as the pili. Okay, they actually hold during mating and all that. Okay, they hold two bacterial cells together and allow DNA to be transferred. How, as I said, plasmid gets transferred, right? So that kind of plasmid transfer process can be carried out by the pili or pilus. Okay, so this was pili and fimbriae. The last thing which you can see is the flagella. So flagella is basically just one of them will be there. Mostly you can find only one flagella. And this is one of the most important appendage for movement. So what it does, it is actually a thick whip-like structure. We have whips, right? No, whip. Okay, so they are whip-like structure. And what they do, they use a propeller kind of motion. They turn it and try to move. They turn their flagella and move forward. So it will look, suppose this is the flagella, it will turn and push the body above. Again, it will turn and push the body above. Okay, so that is how the flagella works. And it is actually it is attached to mostly the cell membrane, not even the capsule or the wall. No, it is very deeply attached with the cell membrane, and it breaks through from the cell, from the capsule and the cell wall. It breaks through from that region. Okay, and it is attached here, this body where it is attached, it is known as the basal body. And as I said, they are involved in the movements of the prokaryotic cell. Okay. Okay. So that's it. Okay. Now, if you have any other doubts, or anything regarding the different parts of the prokaryotic cell, please let me know in the comments. We learned a lot of things. I'll just recapitulate what we learned. Okay, so we started with the discovery of cells. We learned about Roger Bacon, we learned about Zacharias, okay, and Jansen and Robert Hooke and everything. We learned about the different kinds of microscopes, cell theory, and the different kinds of cell, unicellular, multicellular, the shape, irregular shape of the cells, okay, the different size of cells and different types of cells. Okay, then we learned about the different components of the prokaryotic cell. We learned about the cell capsule, the cell wall. We learned about gram staining, what is staining and everything. We learned about the plasma membrane, and I told you plasma membrane, we'll be discussing more about it in the eukaryotic cell. Okay, then cytoplasm, we discussed about ribosomes, the genetic material, plasmids, and the appendages. If you have any doubts, please let me know and we'll try to solve some questions. Okay. So what is the name of the region where where double-stranded single circular DNA, okay, is found in the prokaryotic cell? As I said, prokaryotic cell does not have true nucleus. Can anybody tell me what is the term which is used for double-stranded DNA, the genetic material in prokaryotic cells? Will anybody try? Okay, that is nucleoid. Okay, it doesn't have a true nucleus, proteonucleus and true nucleus and nucleoplasm are not there. It is nucleoid. This is the genetic material of prokaryotes. Okay. Next thing, in prokaryotic cells, ribosomes are what? Ribosomes are protein synthesizing organelle. Okay, they synthesize protein. Okay, and they are 70S. Okay, and they can be divided further into 30S smaller subunit and 50S larger subunit. Okay, next, in prokaryotes, hair-like outgrowths with attached to the surface of other bacterial cells. Okay, we know three kinds of appendages they have. They have the flagella, they have the pili, and they have the fimbriae. Okay, so pili does what? Pili attaches to other bacterial cells, so that there can be an exchange in the genetic material. So it is the pili which helps for genetic material transfer. Okay, so that is pili. Next, who was the first person to see cells under microscope and give them a name? Will anybody try? This is one of the most important and one of the most easiest question. It is Robert Hooke. Okay, Anton van Leeuwenhoek, he actually saw a living cell, not a dead cell. He actually saw a living cell. And Schleiden and Schwann, they actually found the cell theory. So it was Robert Hooke who was the first person to see a cell, even though it was a dead cell. First person to see a cell. That's all. Okay. So thank you everyone for joining tonight. Okay, I hope you understood even a little bit. You can always go back to the videos and see, okay, what there is and what are the different kinds of prokaryotic cells. You can even go back to the older videos where we have discussed about so many things. We have discussed about different kinds of insects. We have discussed about frogs, cockroaches, etc. We have discussed about tissues, plant tissues, animal tissues. You can actually browse the channel and you can see a lot of different videos about, if you want to know about biology, there are chemistry videos as well and everything. Okay, so don't forget to like, share and subscribe to our channel. Okay, I'll see you guys in the next class. That is the Friday 7 p.m. Okay, bye bye.